Welcome to AP Biology. I'm Gail Pfizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. Today, the topic we are covering is one that colleges have asked that all AP students study, even if it will not be on the exam. Colleges are supporting AP students this year by allowing this more flexible approach to the exam, where it will focus on a lesser amount of content than in other years. But in turn, they have asked that AP students learn today's material. To make this relevant to your upcoming exam, we'll also include some exam prep in today's lesson. Soon, after we've covered some of these key outstanding topics, we'll be posting the full schedule of review topics for the remainder of this course, so that you can choose which topics you'll want to prioritize for review and thus which classes you'll want to attend. So now for an overview of our lesson today. Today, I'll be introducing you to Evidence of Evolution, Topic 7.6, Common Ancestry, Topic 7.7, and Continuing Evolution, which is Topic 7.8. You should already be familiar with mechanisms of evolution, natural selection, mutations, genetic drift, and gene flow, and describing, explaining, identifying, constructing hypotheses, and predicting. By the end of this lesson, you will also be able to explain how morphological, biochemical, and geological data provide evidence that organisms have changed over time. Describe the fundamental molecular and cellular features shared across all domains of life, which provide evidence of common ancestry. Explain how evolution is an ongoing process in living organisms, and use the following science skills: argumentation, questions and methods, and representing and describing data. I hope you have a pen and pencil and a paper and calculator, and if you have graphing paper, that would be great. All right, so for the warm-up. The warm-up is the homework from topic 7.5, which was the previous lesson to tonight, since we're 7.6, 7, and 8. I'll go ahead and, if you're just joining us for the first time, I'm going to read it, and then I'll give you a minute, and hopefully those that watched the last lesson have their homework ready. All right, evolution is one of the unifying themes of biology. Evolution involves change in the frequencies of alleles in a population. For a particular genetic locus in a population, the frequency of the recessive allele, little a, is 0.4, and the frequency of the dominant allele, big A, is 0.6. So A is going to ask us two questions. What is the frequency of each genotype, big A, little a, little a, in this population? And what is the frequency of the dominant phenotype? B, how can the Hardy-Weinberg principle of genetic equilibrium be used to determine whether this population is evolving? And C, identify a particular environmental change and describe how it might alter allelic frequencies in this population. I'm going to give you a moment if you're new to our videos, and if you have your homework ready, you can go ahead and start reviewing that, and I'll show you the answer in just a moment. And let's make it 45 seconds. About 10 more seconds. Okay, now I'm going to show you my homework because Ms. Kate assigned you homework, and I do homework with my students all the time. When they write FRQs, I sit down with them and I write those FRQs mainly because I enjoy it, but I want to be in the moment with them just as much as possible. So this is my homework. I started by reading the questions, and as I'm reading the questions, I'm writing down information that will help me stay organized in my thoughts along the way. So the text told me that the recessive allele, the little a, okay, it was little a, and so I know that is my Q, which is 0.4, and then my dominant allele is big A, and my P was 0.6. I know my equations, P plus Q equals 1, and then I also know that P squared plus 2PQ plus Q squared equals 1. So if I just substitute in those values, I will have, for P squared, I have 0.6 squared plus 2 times 0.4 times 0.6 for my 2pq, and then plus 0.04 squared for my q squared. Now, what I get is a frequency of 0.36 for big A, 0.48 for big A, little a, and 0.16 for little a, little a. Now, if I was dealing with a population of 1,000 people, that would mean that 36% of the population was big A, so that would be 360 people. And if it was for big A, 480 of those 1,000 would be heterozygous, and then 160 of them would be recessive. Now, the second part of that question asks for a dominant phenotype. So I have 0.36 is my big A, and 0.48 is my big A, little a. So I have a 0.84, which is 84% of the population would have the dominant phenotype. Now, I wrote a very simple answer for B and C, and I'm not going to take you through the rubric on this. I'm going to show it to you and show it where it is, so you can go back and look at. But it's very busy, and it's an older style, and I don't want to spend 2 minutes on it. But I put it in so you can look at it later, and you'll get a glance at it in a second. So B, I said if the allelic frequency changes, then evolution has occurred. And for C, if environmental selection pressures, and see, when I first wrote it, I did not put down words that I wanted to put in there. And so I came back and I'm kind of squeezing things in, and that's okay. So if environmental selection pressures change against a recessive allele, then that would mean that natural selection was occurring. So this is the rubric. See what I was talking about? Very, very busy. And so if you want to go, and our numbers are correct, and if you want to go down and read all the other options and strategies for B and C, you can do that at your leisure. All right, the framework of the AP Biology course. We're in the evolution unit. We're in natural selection. The unit is actually called natural selection. Our topic is evidence of evolution, common ancestry, and continuing evolution. Each topic has questions called the learning objectives. That is what we need to be able to do. And we need to know content in order to answer these questions, and that's called our essential knowledge. And each topic also has an associated science skill. This is how we're going to show that we know the answer to the question, which is the learning objective. The science skills that are associated with our topics are representing and describing data, argumentation, and questions and methods. And today we will focus on a topic skill pair of evidence of evolution and representing and describing data. So what do you need to know to do this? So some examples of types of data that provide evidence that organisms have changed over time are geological, morphological, and biochemical. Here's some geological evidence. It can provide evidence for evolution. And okay, so radioactive decay can be measured to determine the age of rocks. The length of time required for the atoms to change into another stable element is called its half-life. And iron interacting with oxygen can indicate where levels of oxygen were or are available. And cyanobacteria produce the earliest fossil evidence in stromatolites in 3.5 billion years ago. And if you haven't seen stromatolites, these are stromatolites. All right, fossils. The age of fossils can be determined by the age of the rocks where a fossil is found, which are called index fossils. I wore my dinosaur dew necklace for this occasion. It's called coprolite, and I have a big rock in my classroom, which I can't get to right now, but I have which is actually a fossil of dinosaur waste. And then I had this polished version on a necklace. All right, so homologies, similar morphology shared by a common ancestor. So homologous means the same set of bones organized in a similar way. So if you look at this human arm, okay, you have your humerus, you have your radius, your ulna, your carpals, your phalanges. And if you look at this dog, bird, and whale, there's the same organization pattern in those bones. And they function in a similar way. All right, vestigial structures. So vestigial means reduced or obsolete function, and some examples are bone structure for legs and some snakes, and the hip bone structures in whales. And in this particular diagram, you'll see that there says it's a mutant male, and he has vestigial wings. Well, those wings will not allow him to fly, yet he still has wings. Okay, fundamental molecular and cellular features are shared and conserved across all domains of life and provide evidence of ancestry. Structural and functional evidence supports the relatedness of organisms in all domains. Organisms use the same biomolecules. Organisms have the same DNA codes for the same 20 amino acids, and conserved protein sequences of amino acids have a distinct function in different organisms. So I'm going to help you process some information that's actually going to help you tomorrow or the next lesson, which is 7.9, and that has to do with phylogeny and phylogenetic trees. And so this is a table that kind of will help you grow towards being able to do that. So here we have some evidence from data supporting evolution, and this is biochemical because we're talking about amino acid differences. So this is a table, and if you'll look, there's in the first column, there's a species 1, 2, 3, 4, and 5. And then across in that row, there's species 1, 2, 3, 4, and 5. Now, you can either work this as from a row with the most information in it or a column. I'm a column person, so I'm going to tell you how I process this information. All right, so I look at species 5, and I can see that it is 5 amino acids different than species 1. Okay, that's 5. And then the next value is 20, and I can see that I am 20 amino acid differences away from 2, and 10 away from 3, and 12 away from 4. So I saw something right here that these 2, 3, and 4 are almost equal in their distance away from species 5. So here we have, if I'm going to give you a second, I want you to think about which one would be the closest relative and which one would be most closely related, and then which one would be the farthest away and which 2 go in the middle. And I'll give you just 10 seconds to think about that, maybe 5. Okay, so the more amino acid differences among the 5 different mammal species means the more time that has passed since their divergence. And the divergence means there was a common ancestor and mutations happened, and these began their journey to becoming a different species, okay? So if you look, this would be the closest one. There's only 5 amino acids different between 1 and 5, and between 2 and 5, there was 20. So this one's the farthest away, and 10 and 12 will be somewhere in the middle. And that's from just some data on a table in that organization is what I wanted you to see. All right, here's another representation of, has to do with both actually DNA nucleotide sequences and protein amino acid sequences, because it involves both, because you wouldn't have that amino acid sequence without that DNA sequence. So let's look at our first column. We have species 1, 2, 3, and 4. And then at the top, it says relative amino acid position. So if this was a polypeptide, this would be amino acid 1, amino acid 2, 3, 4, and so on. So if you look at amino acid 1 in each of the species, 1, 2, 3, 4, they all have valine, okay? If you look in amino acid position 2, everything is histidine. And then 3, leucine, 4 is valine. Oh, but there's something different. There's one amino, keep that in mind. There's one amino acid different there in species 2. And then we have glutamic acid, and then arginine, glutamic acid, then tryptophan, histidine, alanine, valine, cysteine. Something's going on in 4 that's totally different than everything else. So let me give you another view. So this is called a sequence alignment. This is a very elementary sequence alignment. You can, once you proceed in biology, you'll start using the National Center for Biotechnology, and there's alignment tools that you'll be using, and it's quite interesting. Okay, so let's look at, there's valine, histidine, leucine, and then we have that one different amino acid. So what happened there? There was another amino acid coded for, so there probably was a single point mutation that occurred there. Now in position 5, we all of a sudden have arginine, and then we have tryptophan. Everything all of a sudden is not the right amino acid. From position 4, and then starting in 5. So what I think happened there must be a frame shift, and we'll talk about that in the review period in the next videos that are coming up in the coming weeks. And then in the other one, all of a sudden we dropped 2 amino acids. So all of a sudden it stopped before it was supposed to. So then we have nonsense. So if we had to pick one that was the closest related, it would be the one with the least amount of change, the least amount of differences in amino acids. So 2 would be a closer relative to 1 than 3 or 4, if we were looking at that from that perspective. Okay, so let's talk about membranes. They are evidence of common ancestry also, and this is, I'm going to talk about endosymbiosis, and I'm also going to tie in endomembrane system at the same time. So here we have a prokaryotic cell. This cell, this call this cell, though, this will be cell 1. All right, so we have prokaryotic cell, and we can see that there's not a nucleus, but we have a circular, one circular chromosome in the middle, okay? And then over here, in the next, which would be cell 2, we have this invagination of this membrane coming in, and that looks like it's going to be forming a nucleus, a nuclear membrane around, right? And so it eventually will. And then that will also have a connection with the endoplasmic reticulum, vesicles, Golgi apparatus, more vesicles, and the plasma membrane. All of those are made out of these same plasma membrane materials. Okay, now if we look at cell 3, we have our nucleus, right? And now we have this engulfing of this prokaryotic cell that has these respiratory functions, and then it's coming into the cell. And now in the next cell, we have an engulfing of a prokaryotic cell that has photosynthetic functions. And now both of them are in the cell, and now they are going to be functioning as a mitochondria and a chloroplast, okay? Now, so you can wrap your head around this double membrane stuff that I'm about to talk about. I drew this thing at this visual at the bottom. So if this was a cell, and it was engulfing this other cell, which is, this would be this cell right here, and then this would be the prokaryote. Notice that this plasma membrane of that first cell would be around there, and then this would be the, and then this would be the cell, the plasma membrane of the prokaryote cell. So now it has 2 membranes, a double membrane. All right, so evidence supporting the theory of endosymbiosis, double membrane of mitochondria and chloroplasts. Mitochondria and chloroplast size are the same as prokaryotes. Mitochondria and chloroplasts have their own DNA, and mitochondria and chloroplasts replicate through binary fission. All right, evolution is an ongoing process in all living organisms. Population of organisms continue to evolve. An example would be the finch beaks of the Galapagos Islands. Genomic changes occur over time. There is continuous change in the fossil record. Organism populations evolve and gain resistance to antibiotics, pesticides, herbicides, and chemotherapy drugs. And then pathogens evolve and cause emergent disease. This is currently happening as the virus that causes the illness COVID-19 is mutating. So let's talk about basic antibiotic resistance. So let's say that these are bacteria in your body, the little nuts and little snowflakes, okay? And this particular antibiotic will kill the bacteria, but it's only killing those that are sensitive to it. There are some bacteria that are resistant to this antibiotic. So then that allows the resistant bacteria to reproduce, and then eventually they can reproduce and over, and this antibiotic will not kill them. And then they can also give this resistance to other bacteria through conjugation and transduction. All right, so let's talk about, just went out of content, and now we're going into our science skill. So we're going to describe data from a table or graph, including identifying specific data points, and some task verbs that we're going to use today: support, describe, explain, calculate. There are a couple more. There are many in what I'm about to talk about. All right, so here's another table that I wanted to familiarize you. I can't even say that. All right, so we're looking at a table, and it has cell wall composed of cellulose, vascular tissue, roots, seeds enclosed in fruit, and flagellated sperm cells. Now across the top, we have green alga, moss, spruce tree, and apple tree. So this is using symbols that we haven't used before. We've been using percents difference or number differences, and now we have plus or minuses. So let's read our text. Students analyzed several photosynthetic eukaryotic organisms in the laboratory to determine their phylogenetic relationships. The organisms were a spirogyra, a green alga, a moss, a spruce tree, and an apple tree. Table 1 compares several characteristics in the organisms. A plus indicates the trait is present, and a minus indicates the trait is absent. Okay, let's read the question. Which of the following rows of data listed in Table 1 best supports the possibility of a common ancestor for the organisms listed there? And we have choices A, vascular tissue, B, seeds enclosed in fruit, C, cell wall composed of cellulose, and D, flagellated sperm cells. So which of the following rows of data listed in the Table 1 best supports the possibility of a common ancestor for the organisms listed there? So which row? And these are rows, okay, going across. I'll give you just a minute to think about it. Okay, and I hope you chose C, cell wall composed of cellulose. If you'll notice, each of these have this trait. So they are, I'll let Ms. Kate talk to you more about this in the next lesson, but they all have this trait, and then only 2 of them have this trait, and 2 of them have that trait. So there's some divergence that's going to happen that they all don't have those traits, okay? All right. More of that to come tomorrow. All right, so now let's look at this data represented in another way. The data below summarize the number of differences in amino acid sequences of cytochrome C found in selected species. So if we're looking at humans to chimpanzees, there's 0 differences in amino acid sequences. That's what we're talking about in this particular protein. And humans have 1 difference from rhesus monkeys in this particular cytochrome C, and in between humans and horses and donkeys, there's 7. Humans and cows or pigs or sheep, there's 7. Humans and rabbits, 7. Birds and reptiles, 10 to 15. Fish, 18 to 20. Plants, 45 to 48. Okay, interpretation of the data supports which of the following statements? Okay, follow along with me. So rhesus monkeys are more closely related to rabbits than they are to horses. So look at where they are. Here's rhesus monkeys, and there's rabbits, okay? And there's horses. So are they more closely related to rabbits? No, because they're the same. But that doesn't mean it's the same changes in amino acids. It just means the amount of that's the same, okay? All right, horses and cows have identical amino acids sequences in their cytochrome C. Okay, so the horses and cows both have 7, but we don't know that which 7. It doesn't mean it's the same 7. Humans are more closely related to rabbits than they are rhesus monkeys. So humans to rabbits, 7. Humans to rhesus monkeys, 1. So humans are more closely related to rabbits. No. All right, plants and animals have no similarities at all. Yes, they do, because if there are 45 to 48 differences, that means that there are similarities also. And mammals are more closely related to reptiles than they are fish. We have mammals to reptiles, there's only 10 to 15 differences, and mammals to fish, there's 18 to 20. So we are more closely related to birds and reptiles than to fish. So the answer to that is E. Okay, so now let's make sure that you understand how to use these tables with this one multiple choice question. You have species 1, 2, 3, 4, and 5, and you have columns, and you have a crazy looking titles in each where the E's, it's all different. Anyway, hemoglobin is a highly conserved protein used by all mammals to transport oxygen. Table 1 was constructed by comparing the amino acid sequence in the hemoglobin molecules from 5 different mammal species. Okay, so what do we know how to do? I'm going to give you I'm going to give you a minute to think about it. Oh, let me read it to you. Which of the following best describes the importance of the number of amino acid differences indicated in Table 1? A, they reflect the different environments where the organisms were captured. B, they reflect the number of base pair changes in the DNA. C, they reflect random changes due to translation errors. And D, they reflect the number of oxygen molecules each hemoglobin molecule carries. Okay, I hope you chose B. All right, let's look at those answers. They reflect the different environments where the organisms were captured. This table doesn't tell you anything about an environment, okay? So not the answer. They reflect random changes due to translation errors. No, that would be a better choice if it had said maybe replication errors. All right. They reflect the number of oxygen molecules each hemoglobin molecule carries. No, it's not about that at all. It's about amino acid differences. So how do we code for an amino acid? It has to do with the DNA, okay? It has to do with those 3 letters of the 3 nucleotides that are going to be the codon for that particular amino acid to become part of that polypeptide. All right, so before I flip this, I don't want you to freak out when you see this, because this is what kids do as soon as they see something that looks massive like I'm about to show you, okay? All right, here we go. Oh my gosh, right? It has writing. There's so much text. I have to read through this. So let's take this in baby steps. I want you to not be afraid of a question that looks like this. So first thing I do is, where's the table? Where's the graph? I've got to have something to look at first. So I'm going to flip this page here. And so here I have a table. It says, and I'm going to assume the role of a student right now. All right, table 1, IC50. I have no idea what that is. Okay, for common anti-malarial drugs tested on artesunate sensitive, oh my gosh, more big words, ARTS and artesunate resistant, ARTR, plasmodium strains. And then I see there's drugs, amodiaquine, chloroquine, DHA, flefaquine, and quinine. And then I see we have some data. We have some ARTS and we have some ARTR. And we had the IC50 again, and there's some plus or minus. Okay, something I can recognize, right? You have your SEM. So can I just do this if right now I'm assuming the role of a student that does not know what IC50 is and can't decipher what these ARTs are? I can't do this with just looking at this table. So I'm going to have to go back and read that text. And I'm going to have to, sorry about that, I'm going to have to engage in reading this much text. And as I'm going, I'm writing myself any notes that I need to carry with me so I don't have to reread it. Okay, but read it good, read it and engage in it. And so you don't have to reread it. All right. Malaria is a disease that is caused by the eukaryotic parasite Plasmodium and is transmitted between individuals by the bite of infected mosquitoes. Different species of Plasmodium cause different disease in different vertebrate species, but in all cases, the parasite infects and destroys red blood cells. The repeated emergence of drug-resistant strains of Plasmodium parasites responsible for human infections has caused researchers to continually search for new drugs or combination of drugs that can provide an effective treatment. A drug that has now proven effective in recent years is artesunate, but now artesunate-resistant Plasmodium strains have also appeared. Researchers investigated whether artesunate-resistant Plasmodium strains could be effectively treated by other drugs. All right, so they're looking for something's not working anymore, and they're looking for something else. All right, in a mouse model of malaria, the researchers determined that a particular strain of sensitive Plasmodium, and now we can refer that to as ARTS, could be largely eliminated from the mice at a dose of 0.15 milligrams per kilogram. By repeated exposure of the artesunate-sensitive strain, the ARTS, to increasing concentrations of artesunate, the researchers developed a Plasmodium strain that is highly resistant to artesunate, ARTR, and requires a minimum dose of 240 milligrams per kilogram to kill the resistant, to eliminate parasites. Okay, compared to the 0.15. All right, so the researchers then compared how effectively several common antimalarial drugs eliminate the ARTS and the ARTR strains in vitro. The researchers infected human red blood cells with the ARTS and ARTR plasmodium strains and incubated the infected cells in tissue culture medium. They added serial dilutions of the drugs to replicate cultures and determined that the IC50 for each drug, the concentration of the drug that reduced the parasite load in the red blood cells by 50%. Okay, so what I know to take forward to this table with me is that ARTS, that's my sensitive ones. ARTR, that is my resistant strains. And then with IC50, that's what I need. That's the dose that it takes to kill half of the resistant strain. Okay. Now let's look at the table. And now we can see that we have different values ranging from 11 to 50 in our sensitive strain. And then look at this, ranging from 39 as our lowest point to 216. Oh, look at that SEM, 81. So when you get to graph, that's going to become important, what those numbers, the range of those numbers. All right, so let's read first what A wants us to do. Plasmodium is an organism with both sexual and asexual life cycles. Describe the most likely cause of heritable phenotypic change in some members of the population of asexually reproducing organisms. Explain how repeated exposure of the artesunate-sensitive Plasmodium strain to increasing concentrations of artesunate led to the development of the artesunate-resistant Plasmodium strain. Okay, so there's two things you need to do. Describe the likely cause of this phenotypic change, which is heritable from one to another, one offspring to next generation, and explain how then it keeps it's developed, how you go from some resistant to a population of resistant. Okay. I'll give you, let's do a minute and a half on that. I'll tell you when you have about 15 seconds left. Okay, let's stop in about 15 seconds. Okay, your describe answer should be similar to this. The response indicates that the most likely cause is random mutation. In parentheses, it says, however, differential gene activation in some organisms under different environmental conditions is also possible. But primarily, most people would have answered the response indicates that most likely cause is random mutation. Okay, so they explain. The response indicates that each increasing concentration of artesunate selected for only those members of the artesunate sensitive Plasmodium population that could survive and reproduce, and eventually only the artesunate resistant organisms remain to reproduce, just like in the Petri dishes that I showed you a few minutes ago. All right, now we're going to graph the data on that table, and then we're going to answer a question about it. So let's go ahead and let's take the table, let's do the table first and graph it. And I'm going to go back and bring up the data again. And then I'm going to tell you a little bit as you graph that, okay? So don't just jump in. Well, first of all, if you have graph paper, you possibly have a whole sheet. And just don't jump in and start throwing numbers on there. Put your eyes on this table and look and see what your range is going to be. And go ahead and start thinking about what is the results, okay? These are results in these two columns. So if these are results in these two columns, those are going to be on your y-axis, okay? Because that's the dependent variable. So when you label, make sure all of this, because this is your results and this is your units, that it goes with you, okay? And then you're going to have to also say and, because you're going to put all of this on one graph. So put all of this information as your label on your, and don't be lazy about it, and just write it all, okay? All right. And then you have on your X, so what you're changing, what's the variable here, is that you're changing the drug, okay? So you can write drugs, and then you can put the amodiaquine, chloroquine, DHA, methylquine, and the quinine all on the X-axis. But when you start scaling, look at this. This is your lowest value, 11.3, and this is your highest value, 216.9 is 81. What is that? 297, 298. You're going to need a range from 0 to 300 to pull this off. So scale correctly. You can't jump around and make different rows worth different amounts, okay? So go ahead and start that. You need that back. Okay, so I'll give you a minute and a half to at least start your graph. You won't be able to do the whole thing because that's going to take some time, but you can come back and look at this video and do it again, because that's what my kids in my classroom will be doing tomorrow morning. You should be graphing. Okay, I'm going to go ahead and stop you. And then we're going to take a look at my graphing, okay? So here's my scaling. There's my 0 to my 300. That's where I started. And then I took a look at how much room I had on my X, and I know I had 5 drugs involved. You can look really closely. You can see my little dots of how I want this graph to be proportional and look nice when I get finished, so it's easy to interpret. All right, so I started, and then I finished with my labeling. See, here's what I was talking about. Everything is over there, okay, on my y-axis, and then everything is here on my X. And then because I'm extra sometimes, I actually put the title on. You wouldn't have to do that on an AP exam, but sometimes if this isn't real clear and you set it up here, then it might score you that point. All right, and then I added the data, and I added my error bars. And so I actually, you don't have to do this, but I do this just for clarity, is there's my mean values, okay? And then I took my mean plus my like 132.4 plus that 16.9, that brought me to 150. Now I'm not going to do the math for each side of this because I have a pretty good eyeball on being even, but you can just subtract 16.9 to get the other side. Now, excuse me. All right, so here's the second part. So we have there's a sensitive and resistant of each of the drugs. So based on the data, determine the antimalarial drugs that are likely to be significantly less. So I'm using that significantly less effective word, okay, at treating disease caused by the ARTR strain than by the ARTS strain. Okay, so now let's look at our error bars. That's going to help us decide which of these 3 are less effective on the resistant, okay? That's what it's asking for. Take a few seconds to do that. Have you chosen 3? Okay, so we're looking for less effective, and so that would be that look at this. Look at this quinine. We have error bars not overlapping, and look at the difference in the two. It takes all of this value to do the job, okay, than it does when the in the sensitive strain. So this is one, and let's see, we have these two. We have some overlapping error bars, so there's nothing significant going on there. Yes, here on the DHA. No on the chloroquine, and yes on the amodiaquine. All right, so when you're graphing, make sure you have your axes labeled, and your scaling has to be right, your units have to be right, and all of your data plotted correctly. All right, and then the labeling, so that should have been on each side, and we did a good job on that. We use the error bars, and we have that amodiaquine, DHA, and quinine have significantly higher IC50s for ARTR than ARTS, and are therefore likely to be less effective at treating disease by the ARTR than the ARTS. All right, so let's go over what we did today. Takeaways for today: geological, morphological, biochemical, and mathematical data provide evidence of evolution. You should also be able to describe the fundamental molecular and cellular features shared across all domains of life, which provide evidence of common ancestry. Explain how evolution is an ongoing process in all living organisms. Use the following science skills: argumentation, questions and methods, and representing and describing data. Common errors and misunderstandings. I kept this brief because I knew I'd be running out of time. Don't lose points with your graphing. That's 3 points that you can, there's no reason to lose those. Don't be lazy with your labeling, okay? Put the units there, put everything there, okay? Just don't leave anything behind. And then your error bars, you need to be accurate above and below, your data points in the right place, and scaling. You can't jump around. You can't say all of a sudden this one is 10 and then the next one is 50. Can't do that. All right, but don't leave those 3 points behind. And here's your homework for tonight. So very short FRQ, and we're going to use the word describe, and we're going to talk about endosymbiotic theory. According to the endosymbiotic theory, some organelles are believed to have evolved through a symbiotic relationship between eukaryotic and prokaryotic cells. Describe 3 observations that support the endosymbiotic theory. You can leave this behind, and when you can look at the video later to look at it again, and then start writing that for tomorrow night's warm-up. All right, device and internet access. We know that not all students have access to the internet or device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity, or know someone who does, you can reach us directly to let us know at cb.org/tech. And thanks for watching, and I hope you come back for the next lesson, which will be 7.9 phylogeny with Ms. Kate. Be sure to do your homework, okay?